So hello everyone and welcome back to Neetopedia, that is Neetka Encyclopedia, and we are back with our biology audiobooks on plant physiology. Please note that mineral nutrition and transport in plant has already been uploaded on our channel. You can check it out in the channel playlist or just click on that I button of the video and you can find the link there. So without wasting our time, let's begin with chapter 13, Photosynthesis in higher plants. All animals, including humans, depend on plants for their food. Have you ever wondered from where plants get their food? Green plants, in fact, have to make their own or rather synthesize the food they need, and all other organisms depend on them for their needs. The green plants make or rather synthesize the food they need through photosynthesis and are called as autotrophs. You have already learned that autotrophic nutrition is found only in plants, and all other organisms depend on the green plants for food and are heterotrophs. Green plants carry out photosynthesis, a physiochemical process by which they use light energy to drive the synthesis of organic compounds. Ultimately, all living organisms on Earth depend upon sunlight or energy. The use of energy from sunlight by plants during photosynthesis is the basis of life on Earth. Photosynthesis is important due to two reasons. It is the primary source of all food on Earth. It is also responsible for the release of oxygen into the atmosphere by green plants. Have you ever thought what would happen if there were no oxygen to breathe? This chapter focuses on the structure of photosynthetic machinery and the various reactions that transform light energy into chemical energy. 13.1 What do we know? Let us try to find out what we already know about photosynthesis. Some simple experiments you have done in earlier classes have shown that chlorophyll, light and CO2 are required for photosynthesis to occur. You may have carried out experiments to look for starch formation in two leaves, a variegated leaf or a leaf that has partially covered with dark paper and exposed to light. On testing these leaves for the presence of starch, it was clear that photosynthesis occurred only in green parts of the leaves in the presence of light. Another experiment you may have carried out where a part of leaf is enclosed in a test tube containing some KOH-soaked cotton, which absorbs CO2, while the other half is exposed to air. This setup is then placed in light for some time. On testing the presence of starch later in the two parts of the leaves, you must have found out that the exposed part of the leaf tested positive for starch while the portion that was in tube tested negative. This showed that CO2 was required for photosynthesis. Can you explain how this conclusion can be drawn? 13.2 Early experiments. It is interesting to learn about those simple experiments that led to gradual development in our understanding of photosynthesis. Joseph Priestley in 1770 performed a series of experiments that revealed the essential role of air in the growth of green plants. Priestley, you must recall, discovered oxygen in 1774. Priestley observed that a candle burning in a closed space, a bell jar, soon gets extinguished. Similarly, a mouse would soon suffocate in a closed space. He concluded that a burning candle or an animal that breathed the air both somehow damaged the air, but when placed a meat plant in the same bell jar, he found that the mouse stayed alive and the candle continued to burn. Priestley hypothesized as follows that plants restore the air whatever breathing animals and burning candles remove. Can you imagine how Priestley would have conducted the experiment using a candle and a plant? Remember, he would need to rekindle the candle to test whether it burns after a few days. How many different ways can you think of to light the candle without disturbing the setup? Using a similar setup as the one used by Priestley, but placing it once in dark and once in sunlight, Jan Ingenhousz showed that sunlight is essential to the plant process that somehow purifies the air fouled by the burning candles or breathing animals. Ingenhousz, in an elegant experiment with an aquatic plant, showed that in bright sunlight, small bubbles were formed around the green plants, while in the dark they did not. Later, he identified these bubbles to be of oxygen. Hence, he showed that it is only the green parts of the plant that could release the oxygen. It was not until about 1854 that Julius von Sack provided evidence for production of glucose when plants grow. Glucose is usually stored as starch. His later studies showed that in green substances in plants, chlorophyll as we know it for now, is located in special bodies which were later called chloroplasts within the plant cells. He found that green plants in plants where glucose is made and that the glucose is usually stored as starch. Now consider the interesting experiments done by T.W. Engelmann. Using a prism, he split the light into various spectral components and then illuminated a green alga, Cladophora, placed it in a suspension of aerobic bacteria. The bacteria were used to detect the sites of oxygen evolution. He observed that the bacteria accumulated mainly in the region of blue and red light of the split spectrum. A forced action spectrum of photosynthesis was thus described. It resembles roughly the absorption spectra of chlorophyll A and B. By the middle of the 19th century, the key features of plant photosynthesis were known, namely that the plant could use light energy to make carbohydrates from CO2 and water. The empirical equation representing the total process of photosynthesis for oxygen-evolving organisms was then understood as CO2 plus H2O in the presence of light gives CH2O plus O2, where CH2O represented a carbohydrate, for example glucose, a 6-carbon sugar. A milestone contribution to the understanding of photosynthesis was made by a microbiologist, Cornelius van Niel, who based on his studies of purple and green bacteria demonstrated that photosynthesis is essentially a light-dependent reaction in which hydrogen from a suitable oxidizable compound reduces carbon dioxide to carbohydrates. This can be expressed by 2H2A plus CO2 in the presence of light gives 2A CH2O plus water. In green plants, water is the hydrogen donor and is oxidized to oxygen. Some organisms do not release oxygen during photosynthesis. When hydrogen sulfide instead of is the hydrogen donor for purple and green sulfur bacteria. The oxidation product is sulfur or sulfate depending upon the organism and not oxygen. Hence, he inferred that oxygen involved by green plants comes from water, not from carbon dioxide. This was later proved by using the radioisotopic techniques. The correct equation that could be represented by the overall process of photosynthesis is therefore 6CO2 plus 12H2O in the presence of light gives C6H12O6 plus 6H2O plus 6O2, where C6H12O6 represents glucose. The oxygen released is from water. This was proved using radioisotope techniques. Note that this is not a single reaction but description of a multi-step process called photosynthesis. Can you explain why 12 molecules of water as substrate were used in the equation given above? Well, please get an answer to this and pin it down in the comment section. If you don't have the answer, please let me know and I'll let you know it pretty soon. 13.3. Where does photosynthesis take place? You would of course answer in green leaf or in chloroplast based on what you have read in chapter 8. You are definitely right. Photosynthesis does take place in green leaves of the plant, but it does also in other green parts of the plant. Can you name some other parts where it think photosynthesis may occur? You would recollect from previous unit that the mesophyll cells in the leaves have large number of chloroplasts. Usually, the chloroplasts align themselves along the wall of mesophyll cells, such that they get the optimum quantity of incident light. When do you think the chloroplast will be aligned with the flat surfaces parallel to the walls? When would they be perpendicular to the incident light? You have studied the structure of chloroplast in chapter 8. Within the chloroplast, there is membranous system consisting of grana, the stroma lamella, and the matrix stroma. It is clear the division of labor within the chloroplast. The membrane system is responsible for trapping the light energy and also the synthesis of ATP and NADPH. In stroma, enzymatic reactions synthesize sugar, which in turn forms starch. The former set of reactions, since they are directly light-driven, are called light reactions or photochemical reactions. The latter are not directly driven but are dependent on the products of light reactions like ATP and NADPH. Hence, to distinguish the latter, they are called by convention as dark reactions or carbon reactions. However, this should not be construed to mean that they occur in darkness or that they are not light dependent. 13.4. How many types of pigments are involved in photosynthesis? Looking at the plant, you have wondered why and how are there so many shades of green in their leaves, even in the same plant. We can look for an answer to this question by trying to separate the leaf pigments or any green plant through paper chromatography. A chromatographic separation of leaf pigment shows that the color that we see in leaves is not due to the single pigment but due to 4 pigments. chlorophyll A, which is bright or blue-green in chromatogram, chlorophyll B, which is yellow or green, xanthophylls, which is yellow, and carotenoids, which are yellow to yellow-orange in color. Let us now see what roles various pigments play in photosynthesis. Pigments are substances that have an ability to absorb light at specific wavelengths. Can you guess which is the most abundant plant pigment in the world? Well, that's chlorophyll A. Let us study the graph showing the ability of chlorophyll A pigment to absorb lights of different wavelengths. Of course, you are familiar with the wavelength of the visible spectrum of light as well as the VIBGYOR. From figure 13.3a, you can determine the wavelength of light at which chlorophyll shows the maximum absorption. Does it show any other absorption peak at any other wavelengths too? If yes, which one? Now look at figure 13.3b showing the wavelengths at which maximum photosynthesis occurs in a plant. Can you see that the wavelength at which there is maximum absorption by chlorophyll A, that is in blue and red regions, also shows higher rate of photosynthesis. Hence, we can conclude that chlorophyll A is the chief pigment associated with photosynthesis. But by looking at figure 13.c, you can see that there is a complete one-to-one overlap between the absorption spectrum of chlorophyll A and the action spectrum of photosynthesis. These graphs together show that most of the photosynthesis takes place in blue and red regions of the spectrum. Some photosynthesis does take place in other wavelengths of the visible spectrum. Let us see how this happens. Though chlorophyll is the major pigment responsible for trapping light, other thylakoid pigments like chlorophyll B, xanthophylls and carotenoids, which are called accessory pigments, also absorb light and transfer the energy to chlorophyll A. Indeed, they not very enable a wider range of wavelength of incoming light to be utilized for photosynthesis, but also protect chlorophyll A from photooxidation. 13.5. What is light reaction? Light reaction or the photochemical phase include light absorption, water splitting, oxygen release, and the formation of high-energy chemical intermediates ATP and NADPH. Several protein complexes are involved in the process. The pigments are organized into two discrete photochemical light harvesting complexes, that is LHC, within the photosystem 1 and photosystem 2. These are named in the sequence of their discovery and not in the sequence that they function during the light reaction. The LHC are made up of hundreds of pigment molecules bound to the proteins. Each photosystem has all the pigments except one molecule of chlorophyll A, forming the light harvesting system, also called antenna. These pigments help to make photosynthesis more efficient by absorbing different wavelengths of light. A single chlorophyll A molecule forms the reaction center. The reaction center is different in both the photosystems. In PS1, the reaction center chlorophyll A has an absorption peak at 700 nanometer, hence is called P700, while in PS2, it has absorption maxima at 680 nanometer and is called P680. 13.6. The electron transport. In photosystem 2, the reaction center chlorophyll A absorbs 680 nanometer wavelength of red light, causing the electrons to become excited and jump into an orbit farther than the atomic nucleus. These electrons are picked up by electron acceptor, which passes them to an electron transport system consisting of cytochromes. This movement of electron is downhill in terms of oxidation reduction or redox potential scale. The electrons are not used up as they pass through the electron transport chain, but are passed on to the pigments of photosystem 1. Simultaneously, electrons in the reaction center of PS1 are also excited when they receive red light of wavelength 700 nanometer and are transferred to another acceptor molecule that has greater redox potential. These electrons are then moved downhill again, this time to a molecule of energy-rich NADP+. The addition of these electrons reduces NADP+ to NADPH plus H+. This whole scheme of transfer of electrons starting from the PS1 uphill to the acceptor down to the electron transport chain to PS1, excitation of electrons, transfer to another acceptor and finally downhill to NADP+, reducing it to NADPH plus H+, is called the Z scheme due to its characteristic shape. This shape is formed when all the carriers are placed in a sequence on a redox potential scale. 13.6.1 Splitting of water. You would then ask, how does PS2 supply electrons continuously? The electrons that are moved from the photosystem 2 must be replaced. This is achieved by the electrons available due to splitting of water. The splitting of water is associated with PS2. Water is split into 2H+, nascent oxygen and electrons. This creates oxygen, one of the net products of photosynthesis. The electron released to replace those removed from photosystem 1 are provided by photosystem 2. We need to emphasize here that the water splitting complex is associated with PS2, which itself is physically located on the inner side of the membrane of the thylakoid. Then, where are the protons and oxygen formed? Likely to be released in the lumen or on the outer side of the membrane. 13.6.2 Cyclic and Non-Cyclic Photophosphorylation. Living organisms have capability of extracting energy from oxidizable substances and store this in the form of bond energy. Special substances like ATP carry this energy in their chemical bonds. The process through which ATP is synthesized by cells in mitochondria and chloroplast is named phosphorylation. Photophosphorylation is the synthesis of ATP from ADP and inorganic phosphate in the presence of light, where the two photosystems work in a series. First PS2 and then PS1, a process called as non-cyclic photophosphorylation occurs. The two photosystems are connected through an electron transport chain as seen earlier in the Z scheme. Both ATP and NADPH plus H plus are synthesized by this kind of electron flow. When only PS1 is functional, the electron is circulated within the photosystem and the phosphorylation occurs due to cyclic flow of electrons. A possible location where this could be happening is in the stroma lamella, where the membrane or lamella of the grana have both PS1 and PS2. The stroma lamella membrane occurs PS2 as well as Nadp reductase enzyme. The excited electron does not pass on to Nadp+, but is cycled back to PS1 complex through the electron transport chain. The cyclic flow hence results only in the synthesis of ATP but not of NADPH plus H+. Cyclic phosphorylation also occurs when only light of wavelength beyond 680 nanometer are available for excitation. 13.6.3 Chemiosmotic hypothesis. Let us now try and understand how actually ATP is synthesized in the chloroplast. The chemiosmotic hypothesis has been put forward to explain the mechanism. Like in respiration, in photosynthesis too, ATP synthesis is linked to development of a proton gradient across a membrane. This time, these are the membranes of thylakoid. There is one difference though. Here the proton accumulation is towards the inner side of the membrane, that is in the lumen. In respiration, protons accumulate in the intermembrane space of the mitochondria when electrons move through the electron transport system. Let us understand what causes the proton gradient across the membrane. We need to consider again the processes that take place during the activation of electrons and the transport to determine the steps that cause a proton gradient to develop. A. Since splitting of the water molecules take place on the inner side of the membrane, the protons or hydrogen ions that are produced by the splitting of water accumulate within the lumen of the thylakoid. B. As electrons move through the photosystems, protons are transported across the membrane. This happens because the primary acceptor of electron, which is located towards the outer side of the membrane, transfers its electron not to an electron carrier but to an hydrogen carrier. Hence, this molecule removes a proton from the stroma while transporting an electron. When this molecule passes its electron to the electron carrier on the inner side of the membrane, the proton is released into inner side of the lumen side of the membrane. Number C. The Nadp reductase enzyme is located on the stroma side of the membrane, along with electrons that comes from the acceptor of the electrons of PS1. Protons are necessary for the reduction of Nadp plus to NADPH plus H plus. These protons are also removed from the stroma. Hence, within the chloroplast, protons in the stroma decrease in number, while in the lumen there is accumulation of protons. This creates a proton gradient across the thylakoid membrane as well as a measurable decrease in pH in the lumen. Why are we so interested in proton gradient? This gradient is important because it is the breakdown of this gradient that leads to the synthesis of ATP. The gradient is broken down due to movement of protons across the membrane to the stroma through a transmembrane channel of CF0 of the ADP synthase. The ADP synthase enzyme consists of two parts. One called the CF0 is embedded in the thylakoid membrane and forms the transmembrane channel that carries out the facilitated diffusion of protons across the membrane. The other portion is called CF1 and it protrudes out on the outer surface of the thylakoid membrane on the side that faces the stroma. The breakdown of the gradient provides enough energy to cause a conformational change in the CF1 particle of the ATP synthase, which makes the enzyme synthesize several molecules of energy-packed ATP. Chemiosmosis requires a membrane, a proton pump, a proton gradient, and ATP synthase. The energy is used to pump protons across a membrane. To create a gradient or a high concentration of protons within the thylakoid membrane, ATP synthase has a channel that allows diffusion of protons back across the membrane. This releases enough energy to activate ATP synthase enzyme that catalyzes the formation of ATP. Along with NADPH produced by the movement of electrons, the ATP will be used immediately in the biosynthetic reaction taking place in the stroma, responsible for fixing CO2 and synthesis of sugars. 13.7. Where are the ATP and NADPH used? We learned that the products of light reactions are ATP, NADPH and O2. Of these, O2 diffuses out the chloroplast while ATP and NADPH are used to drive the processes leading to the synthesis of food, more accurately sugars. This is the biosynthetic phase of photosynthesis. This process does not depend directly upon the presence of light but is dependent on the products of the light reactions, that is ATP and NADPH, besides CO2 and H2O. You may wonder how this could be verified. It is simple. Immediately after light becomes unavailable, biosynthetic process continues for some time and then stops. If then light is made available, the synthesis starts again. Can we hence say that calling the biosynthetic phase as the dark phase is a misnomer? Discuss this among yourself. Let us now see how the ATP and NADPH are used in biosynthetic phase. We saw earlier that CO2 is combined with water to produce CH2 whole line of sugars. It is of what interest to scientists that find out this reaction proceeded, or rather what was the first product formed when CO2 is taken into reaction or fixed. Just after the World War II, several efforts to put radioisotopes to beneficial use. The work of Melvin Calvin is exemplary. The use of radioactive carbon-14 by him in algal photosynthesis studies led to the discovery that first CO2 fixation product was a 3-carbon organic acid. He also contributed to working out the complete biosynthetic pathway and is called the Calvin cycle after him. The first product identified was 3-phosphoglyceric acid, or in short PGA. How many carbon atoms does it have? Scientists also tried to know whether all plants have PGA as first product of carbon fixation, or whether any other product was formed in other plants. Experiments conducted over a wide range of plants led to the discovery of another group of plants where the first stable product of CO2 fixation was again an organic acid, but one with 4 carbon atoms in it. This acid was identified to be oxaloacetic acids, or OAA. Since then, CO2 assimilation during photosynthesis was said to be of 2 main types: those in which the first product of CO2 fixation is a C3 acid, that is PGA, and is the C3 pathway, and those in which the first product was C4 acid, that is the C4 pathway. These 2 groups of plants showed other associated characteristics which will be discussed later. 13.7.1 The Primary Acceptor of CO2. Let us now ask ourselves a question that was asked out by many scientists who were struggling to understand the dark reaction. How many carbon atoms would a molecule have after accepting CO2? Would have 3 carbons of PGA. The studies were unexpectedly showed that the acceptor molecules was a 5-carbon ketose sugar, ribulose bisphosphate, that is RUBP. Did any of you think about this possibility? Do not worry. The scientists also took a long time and conducted many experiments to reach this conclusion. It is also believed that since the first product was a C3 acid, the primary acceptor would be a 2-carbon compound. They spent many years trying to identify a 2-carbon compound before they discovered the 5-carbon RUBP. 13.7.2 The Calvin cycle. Calvin and his co-workers then worked out the whole pathway and showed that the pathway operated in a cyclic manner. The RUBP was regenerated. Let us now see how the Calvin pathway operates and where the sugar is synthesized. Let us at the outset understand very clearly that Calvin pathway occurs in all photosynthetic plants. It does not matter whether they have C3 or C4 pathways. For the ease of understanding, the Calvin cycle can be described under three cases: carboxylation, reduction, and regeneration. Number 1. Carboxylation. Carboxylation is the fixation of CO2 into a stable organic intermediate. Carboxylation is the most crucial step of Calvin cycle where CO2 is utilized for the carboxylation of AUDOBP. This reaction is catalyzed by the enzyme AUDOBP carboxylase, which results in the formation of two molecules of 3-PGA. Since this enzyme also has oxygenation activity, it would be more correct to call it AUDOBP carboxylase oxygenase or Rubisco. Number 2. Reduction. These are a series of reactions that led to the formation of glucose. The steps involve utilization of two molecules of ATP for phosphorylation and two of NADPH for reduction per CO2 molecule fixed. The fixation of 6 molecules of CO2 and 6 turns of the cycle are required for the formation of the 1 molecule of glucose from the pathway. Number 3. Regeneration. Regeneration of the CO2 acceptor molecule RUBP is crucial if the cycle is to continue uninterrupted. The regeneration steps require 1 ATP for phosphorylation to form RUBP. Hence, for every CO2 molecule entering the Calvin cycle, 3 molecules of ATP and 2 NADPH are required. It is probably to meet this difference in number of ATP and NADPH used in the dark reaction that the cyclic phosphorylation takes place. To make 1 molecule of glucose, 6 turns of cycles are required. Work out how many ATP and NADPH will be required to make 1 molecule of glucose through the Calvin pathway. This might help you to understand all of this if you look at it goes in what comes out of the Calvin cycle. 6 CO2 makes 1 glucose, 18 ATP gives 18 ADP and 12 NADPH gives 12 NADP. 13.8 The C4 pathway. Plants are adapted to dry tropical regions have C4 pathway mentioned earlier. Though these plants have the C4 oxaloacetic acid as the first CO2 fixation product, they use the C3 pathway or Calvin cycle as the main biosynthetic pathway. Then, in what way are they different from the C3 plants? This is a question that you may reasonably ask. C4 plants are special. They show a special type of leaf anatomy. They tolerate high temperatures, they show a response to high light intensities, and they lack a process called Photo Respiration and have greater productivity of biomass. Let us understand one by one. Study vertical sections of leaf, one of a C3 plant, another of a C4 plant. Do you notice the differences? Do both have the same type of mesophils? Do they have similar cells around the vascular bundle sheath? The particularly large cells around the vascular bundles of the C4 plant are the bundle sheath cells. The leaves which have such anatomy have Kranz anatomy. Kranz means wreath and is a reflection of the arrangement of cells. The bundle sheath cells may form several layers around the vascular bundles. They are characterized by having a large number of chloroplasts. Thick walls impervious to the gaseous exchange and no intercellular spaces. You may like to cut a section of leaves of C4 plant, maize or sorghum to observe the Kranz anatomy and the distribution of mesophyll cells. It would be interesting for you to collect leaves of diverse species of plants around you and cut vertical section of the leaves. Observe under the microscope, look for bundle sheath around the vascular bundles. The presence of bundle sheath would help you identify the C4 plants. Now study the pathway shown in figure 13.9. This pathway has been named as Hatch and Slack pathway and is again a cyclic process. Let us study the pathway by listing the steps. The primary CO2 acceptor is a 3-carbon molecule phosphoenol pyruvate or PEP and is present in the mesophyll cells. The enzyme responsible for this fixation is PEP carboxylase or pepcase. It is important to register that the mesophyll cells lack Rubisco enzyme. The C4 acid OAA is formed in mesophyll cells. It then forms 4-carbon compounds like malic acid or aspartic acid in the mesophyll cells itself, which are transported to the bundle sheath cells. In the bundle sheath cells, these C4 acids are broken down to release CO2 and a 3-carbon molecule. The 3-carbon molecule is transported back to the mesophyll where it is converted again to PEP, thus completing the cycle. The CO2 released in the bundle sheath cells enters the C3 or the Calvin pathway. A pathway common to all plants. The bundle sheath cells are rich in an enzyme, ribulose bisphosphate carboxylase oxygenase or Rubisco, but lack peptidase. Thus, the basic pathway that results in the formation of sugars, the Calvin pathway, is common to C3 and C4 plants. Did you know that the Calvin pathway occurs in all mesophyll cells of the C3 plants? In the C4 plants, it does not take place in the mesophyll cells but does so only in the bundle sheath cells. 13.9 Photorespiration. Let us try and understand one more process that creates an important difference between C3 and C4 plants: photorespiration. To understand the photorespiration, we have to know a little bit more about the first step of the Calvin pathway, the first CO2 fixation step. This is the reaction where RuBP combines with CO2 to form two molecules of 3-PGA, that is catalyzed by the Rubisco. Rubisco is one of the most abundant enzyme in the world. It is characterized by the fact that its active site can bind to both CO2 and O2, hence the name. Can you think of how it could be possible? Rubisco has much greater affinity for CO2 when the O2 is nearly equal. Imagine what would happen if it were not so. This binding is competitive. It is relative concentration of O2 and CO2 that determines which of the two will bind to the enzyme. In C3 plants, some oxygen does not bind to Rubisco and hence CO2 fixation is decreased, where RuBP, instead of being converted to two molecules of phosphoglycolate in a pathway called photorespiration, the respiratory pathway, there is neither synthesis of glucose, sugars, nor of ATP. Rather, it results in release of CO2 with the utilization of ATP. In the photorespiratory pathway, there is no synthesis of ATP or NADPH. The biological function of photorespiration is not yet known. In C4 plants, photorespiration does not occur. This is because they have a mechanism that increases the concentration of CO2 at the enzyme site. When the C4 acid from the mesophyll is broken down into the bundle sheath cells to release CO2, this results in increasing the intracellular concentration of CO2. In turn, this ensures that the Rubisco functions as carbolase, minimizing the oxygenase activity. Now that you know that C4 plants lack photorespiration, you probably can understand why productivity and yields are better in these plants. In addition, these plants show tolerance to high temperatures. Based on the above discussion, can you compare the plants showing the C3 and C4 pathway? Use the table format given and fill in the information. So, this table 13.1 is really important from NEET point of view. So please do not forget to fill it out and memorize it for clearing your concepts as well as giving a perfect score in your exams. 13.10 Factors affecting photosynthesis. An understanding of the factors that affect photosynthesis is necessary. The rate of photosynthesis is very important in determining the yield of plants, including crop plants. Photosynthesis is under the influence of several factors, both internal and external. The plant factors include the number, size, age and orientation of leaves, mesophyll cells and chloroplast, internal CO2 concentration and amount of chlorophyll. The plant or internal factors are dependent upon the genetic predisposition and the growth of the plant. The external factors would include the availability of sunlight, temperature, CO2 concentration and water. As the plant photosynthesizes, all these factors will simultaneously affect its rate. Hence, through several factors interact and simultaneously affect the photosynthesis or CO2 fixation. Usually, one factor is the major cause or is the one that limits the rate. Hence, at any point the rate will be determined by the factor available at suboptimum levels. When several factors affect any biochemical process, Blackman's law of limiting factors comes into effect. This states that if a chemical process is affected by more than one factor, then its rate will be determined by the factor which is nearest to its minimal value. It is the factor which directly affects the process if its quantity is changed. For example, despite of the presence of green leaf and optimum light, CO2 conditions, the plant may not photosynthesize if the temperature is very low. This leaf is given the optimal temperature, it will start photosynthesizing. 13.10.1 Light. We need to distinguish between light quality, light intensity and the duration of exposure to light. while discussing the light as a factor that affects photosynthesis. There is a linear relationship between incident light and CO2 fixation rates at low intensities. At high light intensities, gradually the rate does not show further increase as other factors become limiting. What is interesting to note that is, light saturation occurs at 10% of the full sunlight. Hence, except for plant in shade or dense forest, light is rarely a limiting factor in nature. Increase in incident light beyond a point causes the breakdown of chlorophyll and a decrease in photosynthesis. 13.10.2 Carbon dioxide concentration. Carbon dioxide is the major limiting factor for photosynthesis. The concentration of CO2 is very low in atmosphere between 0.03 and 0.04%. Increase in concentration up to 0.05% can cause an increase in CO2 fixation rates. Beyond this, the levels can become damaging over longer periods. The C3 and C4 plant respond differently to CO2 concentrations. At low light conditions, neither group responds to the high CO2 concentrations. At high light intensities, both C3 and C4 plants show increase in the rate of photosynthesis. What is important to note that is, C4 plants show saturation at about 360 micro per liter, while C3 responds to increased CO2 concentration and saturation is seen only beyond 450 micro per liter. Thus, current availability of CO2 levels is limiting to C3 plants. The fact that C3 plants respond to higher CO2 concentration by showing increased rates of photosynthesis leading to the higher productivity has been used for some greenhouse crops such as tomatoes, bell pepper. They are allowed to grow in carbon dioxide enriched atmosphere that leads to higher yields. 13.10.3 Temperature. The dark reactions being enzymatic are temperature controlled. Though the light reactions are also temperature sensitive, they are affected to a much lesser extent. The C4 plants respond to high temperatures and show higher rate of photosynthesis, while C3 plants have a much lower temperature optimum. The temperature optimum for photosynthesis of different plants also depends upon the habitat they are adapted to. The tropical plants have a higher temperature optimum than the plants adapted to the temperate climates. 13.10.4 Water. Even though water is one of the reactants in the light reaction, the effect of water as a factor is more through its effect on the plant, rather than directly on photosynthesis. Water stress causes the stomata to close and hence reducing the CO2 availability. Besides, water stress also makes the leaf wilt, thus reducing the surface area of the leaves and their metabolic activity as well. Summary. Green plants make their own food by photosynthesis. During this process, carbon dioxide from the atmosphere is taken in the leaves through stomata and used for making carbohydrates, principally glucose and starch. Photosynthesis takes place only in the green parts of the plant, mainly the leaves. Within the leaves, the mesophyll cells have a large number of chloroplasts that are responsible for CO2 fixation. Within the chloroplasts, the membranes are site of light reaction, while the chemosynthetic pathway occurs in the stroma. Photosynthesis has two stages, the light reaction and the carbon fixing reactions. In the light reaction, the light energy is absorbed by the pigments present in the antenna and funneled to special chlorophyll A molecules called the reaction center chlorophylls. There are two types of photosystems, PS1 and PS2. PS1 has 700 nanometer absorbing chlorophyll A P700 molecule at its reaction center, while PS2 has P682 reaction center that absorbs red light at 680 nanometer after absorbing light. Electrons are excited and transferred through PS2 and PS1 finally to NADP, forming NADH. During this process, a proton gradient is created across the membrane of the thylakoid. The breakdown of the proton gradient due to movement through the FO part of the ATPase enzyme releases enough energy for synthesis of ATP. Splitting of water molecules is associated with PS2, resulting in the release of O2 protons and transfer of electrons to PS2. In the carbon fixation cycle, CO2 is added by the enzyme Rubisco to a 5-carbon compound RUBP that is converted to 2 molecules of 3-carbon PGA. This is then converted to sugar by Calvin cycle, and the RUBP is regenerated. During this process, ATP and NADPH synthesized in the light reaction are utilized. Rubisco also catalyzes a wasteful oxygenation reaction in C3 plants, that is photo respiration. Some tropical plants show a special type of photosynthesis called C4 pathway. In these plants, first product of CO2 fixation takes place in mesophyll is a 4-carbon compound. In bundle sheath cells, the carbon pathway is carried out for synthesis of carbohydrates. So that's the end of the chapter. I hope you enjoyed listening to this audiobook. See you again in the next one. Till then, subscribe to the channel and stay tuned. Thank you.